All right. Good morning, everybody. Welcome to another episode of Sunday Morning Science. Very excited to be here. Uh, I've got kind of a new setup that hopefully will be helpful. I've got I can see the live chats over here, and I can see the super chats over there. And yeah, we'll try this out. Hopefully, this will run a little bit more smooth than last couple of times. I'm very excited today because we're going to talk about the evolution of new genetic information. And just kind of while people are trickling in here, I just curious, asking you guys, go ahead and respond in the comments here. How many of you started following Nancy after last week's show? Her stuff is awesome. I I just watched her. She did a live Periscope. She uses Periscope to do live videos. It was on Twitter that I saw her just the other day. She was doing something. She was doing a video on the biodiversity of Ecuador, and I love watching her live stuff. She, being a woman on YouTube or on the internet doing live videos is a lot harder than being a dude. She gets constantly sexually harassed in the comments, and she just, like a boss, she responds to it in really hilarious ways. You should go watch some of her live videos. I thought that was entertaining. She's talking about biodiversity, and she's responding to comments, and she's just like, she's reading through them, kind of mumbling like I do, but not as much as she's reading. And she's just like, yeah, my personal life has nothing to do with the biodiversity of Ecuador. I mean, she just constantly, yeah, she's awesome. Responding to things, being a thick-skinned, really good presenter. She's got a really good personality for this type of work. So anyways, you should definitely follow her, SciBugs, on Twitter. And if you follow her on Twitter, you'll have her, you'll find her everywhere. She posts pretty much everything on Twitter. Anyway, we got quite a few people here now, so let's go ahead and dig in. Let's get started. I'm going to open up my, let me start sharing my screen here. And open my presentation. So today we are going to take a look at this paper, 40 Years of In Vitro Evolution. We're going to be mainly answering the questions, how does new genetic information evolve? This is a question that comes up quite a bit. Just a little reminder here, on the left, you can see my contact information. On Twitter, I am at Stated Clearly. On Patreon, I am patreon.com/statedclearly. And today we're going to do a lot of Q&A. Actually, there's two sections of Q&A today, like through the presentation. It's kind of a more dense presentation. I want to make sure that people are following everything that I'm talking about. Super chats are priority for the questions, because it's awesome. I get you guys, can give me a little tip. Super chat. This is a Sunday morning science chat, so it's kind of like passing around the contribution plate. Super chats are, reminder to be nice to people in the comments. There's a lot of people with different views, and some people who might be angry. Just ignore angry people in the comments. Keep things civil, keep things PG if possible. I will also try to keep things PG. So these are the two big questions that we're going to be talking about today. New genetic information cannot evolve. This is the claim that a lot of well-educated, let me just talk about this for a second here. Over time, as biologists have found more and more super obvious evidence for evolution, creationists who insist on denying the obvious fact that life on Earth evolved, they've had to kind of resort to more and more abstract arguments. in order to continue denying the evidence of evolution. And one of those more abstract arguments is this idea that no new genetic information can evolve. And this is something that we see on, like, seriously every single creationist website, from Answers in Genesis to creation.com to, there's also a website by the intelligent design people called Evolution News. I thought that's clever. They bought the URL Evolution News and they put up anti-evolution propaganda on there. Super clever. But they've gotten several articles about this idea that no new genetic information can ever evolve. They're very animate about this. And then the second big question, or big claim that you hear from creationists today, is that evolution can't happen until you have cells and the genetic code. And cells and the genetic code, of course, are both very complex. And so the argument there is, well, you have to have intelligent design at the start of life because these things are so complex. And there's two people that are making that argument. Perry Marshall is a friend of mine. I've had him on the show, actually. He's one of the people that says that you can't get evolution to happen without the genetic code. And he wrote a book about that called Evolution 2.0. And I like Perry Marshall a lot. Again, he and I are friends, and we've had lots of discussions about this in the past. And then there's a guy going around right now, James Tour. He's a chemical engineer and a creationist, and he's going around on this tour talking about the complexity of the cell and the complexity of the biochemistry of the cell. And he makes this claim over and over and over again, that you cannot get evolution until you have the cell. And because the cell is so complex, there had to be an intelligent designer, at least at the start of all of this. And both of these arguments, new genetic information cannot evolve and evolution cannot happen until you have cells and the genetic code. Both of those arguments have been thoroughly debunked for 40 years now. And we're going to talk about one series of experiments that has debunked both of those claims. And we can read about this in this paper called 40 Years of In Vitro Evolution, which goes over a series of experiments on RNA. RNA can actually replicate and evolve. Given the right circumstances, it can replicate and evolve without any need for the genetic code and without any need of the cell. And we're going to talk about these experiments today. They're really proof of concept experiments. The situations in which these evolve, they're not situations that you would have on the early Earth. These are proof of concept. People who are studying the origin of life now are trying to get similar situations to happen in a context that would be more relevant to the environments that existed on the early Earth. But yeah, for 40 years now, we've had this proof of concept, which is absolutely amazing. But before I dig into that paper, and I wanted to respond to a question that I got last night as I was preparing for this by a YouTube user and follower of the Stated Casually channel and the Stated Clearly channel. His name's Isaac. And he wrote me this fairly long question. But his main question is, how does a beneficial mutation happen? And he goes on to talk about insulin. You know, insulin is this molecule. Hold on. Get a drink of water here, my voice. Insulin is this very specific molecule. It's a protein. So it's a protein that's coded for by one of our genes. We have an insulin gene. And he points out the fact that if this mutates, you know, there's actually lots of ways that a gene can mutate without changing the structure of the protein. So there's a lot of neutral mutations that can happen in the insulin gene. But he says that, you know, if a mutation in insulin occurs that does change the protein sequence, a lot of times the person born with that mutation ends up having diabetes. They're born with diabetes. And so he's like, how can beneficial mutations happen when proteins are so sensitive to mutations like this? And furthermore, he goes on to say, and what happens if two or more mutations are required before the change is beneficial. How is it that beneficial mutations can occur? Well, here we have insulin. This is the insulin protein, and again, insulin is used as a communication molecule. It's between tissues, so different tissues in the body signal to each other with the insulin molecule. And the insulin molecule is extremely preserved, is what we say. It's amino acid sequence, so its protein structure is extremely preserved across evolutionary time. Here on the left, I've got a pig insulin molecule. Here on the right, I have a human insulin molecule, and you can see that they are almost identical. There is only one amino acid difference between the two molecules, between humans and pigs. That means that pretty much every single mutation that has ever happened in this molecule has been bad in mammals. And actually, this is true not just in mammals, but in vertebrates in general. If you were to look at, I don't have the fish protein here, but if you look at the insulin molecule in, I think it's tilapia that the research has been done on, it's almost identical to human insulin. And in fact, it's so close to identical that you can actually use it in the human body and it will work. It'll actually do its job in the human body. So Isaac is right. Pretty much any mutation in this protein is bad, but that's actually not super common for proteins. And many proteins, that is not the case at all. And let's see. Sorry, I got lost in my notes here. Yeah, so for many proteins, that's not true at all. And there's lots of proteins, for example, that we've watched in dogs. In dogs, we have this rapid evolution, right? Since they've been domesticated, we have all sorts of different weird shapes and sizes of dogs. And we can look at the mutations that have caused those differences in phenotype, and we can see the proteins that code for those differences. And a lot of the proteins that can easily mutate are proteins that regulate the expression of other proteins. And also the regulatory regions of a gene can also mutate quite a bit without ruining the protein. So, for example, in insulin, it's very rare for a mutation to survive in the protein coding region of insulin, but there are lots of mutations, lots of differences. If you look at one animal to another in the regulatory region of the gene that codes for insulin, and mutations in the regulatory region of insulin will make insulin either produce more rapidly or more slowly. Different mutations will change the regulation of this protein. And so those types of mutations that change the regulation of a protein, those are the main types of mutations that happen across evolutionary time. I've got a whole video series on the Stated Clearly channel. It's actually just 2 videos, but it's on how does new genetic information evolve. And in that animation series, I give a whole bunch of different examples of mutations that give rise to new traits. And one of the ones that I focus in on is there is the cane toad, which I believe is native to Australia. Don't quote me on that, though. I talk about it in the video. It has a toxin that it produces that messes with a protein called the hold on, gotta look this up in my notes. The sodium potassium ion pump. So it creates a protein that messes with that pump. It trips up that pump, blocks it from being useful in the animal that eats the toad or swallows any of this poison. And in most animals, that will kill them. It'll cause seizures and stuff, and then they'll die if they swallow too much of it. But there's lots of animals that have evolved an immunity to it. Lots of predators of the cane toad that have eventually evolved an immunity to this. And they've done that because this paper right here talks about the specific mutations that can cause this immunity. There's a whole bunch of different types of mutations that can cause this immunity, and they act in an additive function. So if you have one of these mutations, you're sort of immune to the cane toad. You can swallow a little bit of it and at least survive. And if you have multiple mutations, you get to the point to where the cane toad's toxin is pretty much useless against you. You're pretty much invincible to it. And this happens, we now know exactly what types of mutations can do this. Again, multiple different types of mutations can give you a resistance to the cane toad, and they're additive, and they can happen in any sequence. One can happen first, then the other. There's like 12 different kinds that can happen. And this is more typical of the types of mutations that we actually see that give rise to, beneficial phenotypes. It's very common. The second animation that I have on the evolution of new genetic information talks about gene duplication events. Gene duplications are another way in which new genetic information evolves. And I talk a lot about this mutation of a new toxin in snakes. There is a toxin that they have that allows them to cause rapid clotting in their victims. And the clotting toxin, which is expressed in their venom gland. By the way, a snake's venom gland is a modified saliva gland. And that toxin actually comes from a gene that they normally use in their own blood clotting system. So snakes, their blood clots when they get cut, just like ours does. And it actually uses most of the same proteins. This is a protein system, a blood clotting system that first evolved in early vertebrates. Actually, it started evolving before vertebrates were a thing. And our ancient ancestors, Precambrian, this thing started, but it really solidified in early vertebrates. So all fish share a very similar blood clotting system to us. And these snakes share a very similar blood clotting system. And what happened in these snakes is there was a gene duplication in one of the proteins that causes blood clotting. That duplication occurred, and the duplicated gene was inserted in a segment of the snake's genome that allowed it to be expressed in the venom gland. And so these snakes were now excreting this blood clotting protein into their venom gland. And so when they would bite an animal, it would rapidly clot at the site of the bite, and this clot could cause problems. There was so much of this clotting factor that was injected into the body of the animal, it actually helped kill them. And further mutations in the duplicated version of the gene that's now expressed in their saliva gland, further mutations to that made it more and more efficient. And now what happens when you get bit by one of these snakes that has this gene in its venom glands? What happens when you get bit by the snake is you will rapidly clot at the site of the bite, and you will clot so quickly that all of the clotting factors in your entire body will get used up right at the site. You'll get a huge, nasty clot right at the site of the bite. And everywhere else, your clotting factors are so used up that you start bleeding from all the little nicks and scrapes that you have on your body, and the internal nicks and scrapes that you have within your own circulatory system. You'll actually bleed to death internally because of this snake bite. It's crazy. Because you're clotting so fast in one area, you bleed out in the rest of your body. Horrible, horrible way to go. But this is how one of the many ways that we have discovered that new genetic information evolves, new beneficial mutations arise, is you have a gene duplication event, and that duplicated gene then undergoes point mutations. It's free to undergo point mutations because it's no longer crucial for your survival and reproduction, because you now have two genes. So that original gene continues to do its function, and this new gene is freed up to mutate. And that's one way that these highly specific proteins can gain new functions. So There is this paper on the Nature Education website. So Nature magazine is a scientific, the Nature journal is a scientific journal, but they also have this kind of public facing side, their Nature Education series. And on there they'll have authors write to the public. So they kind of simplify their research. There's this wonderful paper on the origin of new genes. And you can read that on there. It does a really good job overviewing the research. I mean, it's kind of a little bit old now. It was written in 2008. So there's a lot of new stuff that we have now, particularly on the evolution of genes de novo. So something that I don't talk about in my video series on the evolution of new genetic information, I don't talk about de novo origination of genes, but what can also happen is we have large stretches of our DNA that are not used for producing RNA or protein, and mutations in these non-coding regions. Some people call it junk DNA. It's not really junk DNA. It's actually it does do stuff, but a lot of it does. Not all of it. Some of it is truly just junk DNA, but a lot of it is used for things. But mutations in this so-called junk DNA can cause a stretch of DNA to start being transcribed into RNA, and that RNA can end up having a useful function. A lot of times it just ends up being kind of noise inside the cell, but a lot of times these mutations can give rise to actual functional sequences. And so that is another way that new genes come about. There's a lot of research on that happening right now because it's not a process that we understand very well. There's a lot of research happening on that right now. If you do a Google search for de novo gene evolution, you'll find a whole bunch of different papers on that. And again, this is new. There's a lot of debate and discussion about how this actually occurs. And but yeah, we do know that it happens. So I want to just kind of field some questions now. I'm looking at the super chat section, and I'm looking now at the just the regular chat as well. Let me stop sharing my screen here. This is the fun part of the podcast where I fumble with buttons. There's me again. Let's see. Looking through your questions. Shannon, who else loves Science Sunday? If you've asked questions, again, I'm re-ask them here and I'll be able to see them. Someone says you should have a co-host. Last time I had Nancy on, and it was really helpful because we could both kind of read questions as we went. That was pretty awesome. But yeah, I should get a co-host, huh? So Isaac asks, it will happen automatically, essentially. I'm not sure what you mean by that. Sorry. Okay, so you're saying a regulatory protein does not need to be turned on to a corresponding junk DNA stretch in order to transcribe the junk DNA into an RNA strand. Okay, so Isaac, yes, mutations in the genome within a stretch of junk DNA, mutations in that can cause it to attract what we call regulatory factors, regulatory proteins. So it's just simple mutations in the actual DNA strand can attract transcription factors. Transcription factors are the proteins inside the nucleus of the cell in eukaryotes that attach to a chunk of DNA and essentially tell the nucleus, hey, this chunk of DNA is important. We should transcribe this into RNA. And those transcription factors, all they do is they bind to specific sequences of DNA. So any mutation that causes a stretch of DNA to look like to attract those proteins will cause that stretch of DNA to start being transcribed into RNA. And that RNA molecule might end up doing something useful. It might end up just being noise inside the cell. It actually might end up being toxic. So the research on the evolution of new genes is looking at that sort of thing. And then further mutations can actually cause that to be to actually be used by the ribosome and read out as a protein. So again, a lot of that research is new, but there's a lot of it out there. So yeah, check out that. The paper that I showed you that kind of gives an overview of all of this research is a good place to start. That Nature paper review, and then you can go from there and find all sorts of research done on this. It's really cool. All right. I think that answers the questions that people had about this. So I'm hoping that everything I just said was followable. If you've been following my website for a while, or my YouTube channel for a while, if you watch the What is DNA video, if you watch the What is the RNA World hypothesis video, hopefully everything I just said made sense. I'm going to kind of review things a little bit because the paper that we're going to go over is fairly complex. You have to have a pretty good understanding of what DNA is, how it works. So let me just share my screen again. Share. Pull this up again. So just a super quick overview. And again, I'm assuming that you've already watched my What is DNA video, or you already know what DNA is and how it works. But in modern cells today, DNA, you could essentially say that it produces RNA. That's not totally true. There's proteins inside the nucleus of the cell that read DNA and produce a chain of RNA that's a copy of a certain stretch of DNA. That chain of RNA then goes into the nucleus of the cell. It connects with a ribosome, and the ribosome attaches to the RNA, reads the sequence in the RNA chain 3 letters at a time, and uses that to create a protein. And it uses, so this, by the way, is a legitimate communication system that has evolved between the genome and the ribosome. And I've actually got on the 8th of October on the Non Sequitur show, which is another YouTube channel, I will actually be doing a debate as to whether or not this is a legitimate communication system and whether or not we should consider it a legitimate code, because we call this the genetic code, right? This here is the kind of the cheat sheet to the genetic code. Scientists cracked the genetic code in the late 60s, early 70s. And we've called it the genetic code since we cracked it. And there are some biologists that don't really understand coding very well that say that this is not really a genetic code. It just is sort of similar to one. It's an analogous to one. I will be arguing that it is really a code. And by code, I mean it is a series of rules that govern a legitimate communication system. And that communication system is the communication that exists between the genome and the ribosome. And Yeah, I will be making a case that this is a legitimate communication system, a legitimate code, and why that is. Um, yeah, so genes give rise to chains of RNA. RNA is then read by the ribosome to produce proteins, a specific shape of protein, by reading the code on the RNA chain. And that's a complicated communication system. And there are a lot of people that, uh, like Perry Marshall, for example, says that that communication system had to have been created by an intelligent designer. This is not the case. We know how communication systems evolve. There are lots of communication systems that have evolved in biology. We just talked about insulin, for example. Insulin is a communication molecule. It's a different type of communication that happens in biology. It's a communication molecule that's used between different tissues in our bodies. But there are also examples of communication systems like the one between flowers and bees, for example, a highly complicated communication system that evolved. And the genetic code, this communication system that we find inside the cell, it is also an evolved communication system. And uh, the evolution was happening before we had the existence of the genetic code. Evolution was already happening. And this paper right here, 40 years of in vitro evolution, which we're about to look at, this paper outlines research that demonstrates that evolution can happen without the genetic code. So, any questions on that before I dig in? And again, if you can do a at stated clearly in your question, it makes it way easier for me to see on the live chat here. There's a lot to read through. Someone says, isn't the communication system of insulin a product of DNA? Yes, it is. So insulin is produced by a gene, which we find in DNA. Within biology, there are communication systems upon communication systems upon communication systems. There's anytime you get symbiosis, so the evolution of two or more organisms working together, you by default get a communication system that evolves between those two entities. And the genes in our genome are communicating with each other. They're cooperating with each other. And so there is communication between them. When we evolved multicellular organisms, when that first started happening, again, we had the evolution of a new communication system between cells. And insulin is an example of that, the communication system between cells. So we see every time there's the evolution of symbiosis, there is also the evolution of communication between the two organisms that are cooperating. Again, a lot of people find it weird to think of genes as organisms, but they really are. They should be thought of as organisms. Evolution at its most fundamental level happens at the level of a gene, and these genes are cooperating with each other, and they really do evolve sort of on their own. In an organism, they're locked in cooperation, so they can't evolve completely independently of each other, but they do evolve, and sometimes they compete with each other a little bit. Sometimes there's all sorts of really crazy things that happen when you follow the evolution of individual genes. You'll see that new alliances are sometimes made within the genome. Certain genes get kicked out across evolutionary time, and all of this is happening automatically just through the process of descent with modification acted upon by selection. It's really, really fascinating to get a really good overview of how that works and how to start thinking of evolution at the perspective of a gene. I highly recommend Richard Dawkins' book, The Selfish Gene. That's where he's the one who really first made the case, not first made the case, but strongly made the case in a way that the public could understand that we should be looking at evolution from the perspective of a gene. He kind of oversold that idea in his book. he was fighting against people that were saying we should look at the group, or we should look at the species for where evolution actually happens. And there actually are legitimate reasons to look at the group and the species. Looking at the group is we don't understand how to look at the group very effectively right now, but there are some researchers that are trying to figure out how to do that better. Anyway, this is kind of an ongoing argument in biology. When is it most appropriate to look at genes rather than organisms, rather than populations? But there are a lot of weird questions that we have in biology that are best answered by looking at the evolution of individual genes. Okay. Brian Stevens says, look up, poke, poke. So, okay, here I'm going, Brian. My definition of communication system is different, he says. You will, if you watch on the 8th, you'll hear what a communication system, how a communication system is defined in by a specialist that study communication. And the genetic code, the communication that happens between the genome and the ribosome, is definitely a legitimate communication system. Someone says, this is off topic, but do you think that modern day scientists jumped the gun when naming atoms atoms? Elementary particles are what the Greeks probably thought of as atoms. Yes. John Dalton thought that the atom was a fundamental particle, and he was wrong. They are not fundamental particles. But yeah, I'm going to be debating Steve McRae, who's also a really cool guy, on the 8th. So yeah, go watch that. That'll be fun. Okay, so let's get to the paper here. Actually, let's not get to the paper quite yet. I need to, before the paper is going to make any sense to people, so many windows, I need to talk about RNA. So hopefully most of you have already watched my video on the RNA world hypothesis, but I'm betting that some of you have not, or if you have, it's been a while, right? So this is a clip from the RNA world hypothesis video. And the RNA world hypothesis is this idea that the first evolving entities, before we had cells, before we had proteins, the first evolving entities were chains of RNA. Excuse me. My voice is waning here. There are lots of reasons for this, which you can learn about by watching my video, What is the RNA World Hypothesis? But RNA is really fascinating because it can actually reproduce and evolve almost on its own. We have to set up a really clean environment for it to reproduce and evolve, and we have to add into that environment some sort of a catalyst that can help it reproduce. And we don't yet know, the catalysts that we use right now they're fairly complex enzymes, they're proteins. And we haven't yet found a catalyst that would have existed on the early Earth. And we don't yet have an environment that would have existed on the early Earth. So we're really just doing these experiments as a proof of concept right now. So we have not demonstrated that the RNA world is how things actually evolved on the early Earth, but we're getting closer and closer to doing that. Well, we're getting closer and closer to demonstrating that the RNA world could have been a way that life evolved on the early Earth. But this segment of the video shows how this replication happens. So you've got a chain of RNA. I'm going to hit play here. It's floating around on the water here. There are free RNA nucleotides in the water. And you see that they will base pair with their partners. And if they base pair with their partners, they form a complementary strand. And if that complementary strand is then bound together, its backbone is sewn together and we need a catalyst to do this, you get an actual complementary strand. And then as the temperature in the water changes, you get the two strands to separate. And then if it cools down again, you get base pairing once more. And so through this process, you get RNA replication. And through this process, mutations can crop up. And because of that, these chains of RNA can change in their shape over time. And in the RNA World video, I show that not only can chains of RNA base pair with replicate through base pairing, but they can also base pair with themselves. And what they'll do is they'll bend and kind of fold and stick to themselves and form these really weird, complex three-dimensional structures. And it's those weird, complex three-dimensional structures that can have functions. They can actually act as enzymes. They can do all sorts of weird things. Excuse me. Drink of water here. They can do all sorts of weird things in their environment, and some of those things can be highly functional. So this paper that we're looking at here, pull that up. 40 years of in vitro evolution talks about experiments that we did on the evolution of RNA in the test tube. We've been doing this for over 40 years now. Really good paper. Just in case you're not really familiar with how scientific papers work, most scientific papers are just people reporting on experiments that they have done. And then every once in a while, someone will write an overview paper like this. You know, a lot of the experimental papers, people aren't really presenting their final conclusions about things. They're presenting their tentative conclusions. They're describing their experiments. And there's a lot of debate about the legitimacy of those experiments and the legitimacy of the conclusions that the researchers based on those experiments. So for the most part, when you read the scientific literature, you're kind of reading the middle of an argument. A lot of people assume that if it's published in a peer-reviewed journal, that means it's legitimate science. That means it's a legitimate conclusion. That's not the case. The scientific literature is where scientists argue their points and their conclusions. But every once in a while, you'll get a paper like this, which is a review of all of the research in a field and all of the arguments that have been made in that field. And they're really cool to read. And this one happens to be particularly easy to read and understand if you have a good understanding of how RNA replicates and evolves. And you can get that idea by watching my RNA World video. So the introduction of this paper just kind of goes over how evolution works in general. Darwinian evolution, descent with modification, acted upon by selection, and it talks about how RNA actually can evolve through this process. And again, RNA can evolve without any need of the genetic code, without any need of the cell. We can get it to evolve in vitro, so in the test tube. Section 2, he talks about the first RNA evolution experiment. And what we did is we found this really crazy virus. Hold on, that's the wrong one. This really crazy virus, you can see it right here. It's called the Q beta virus, and it infects bacteria. And it does not have DNA at all, doesn't use DNA at all. Instead, it just has RNA. What it does is it will infect a bacterial cell. It'll get inside of it. It'll excrete its RNA into the bacteria cell. And that RNA will then it'll attach to the ribosome, and it'll be read, and it'll start making all the proteins that make up this virus. So it'll just rapidly make this virus. It also has a protein that replicates chains of RNA. And it doesn't just replicate any chain of RNA. It replicates specifically chains of RNA that have a special sequence in them that all of the virus genes have. So it only replicates viral strands of RNA. And so the guy that discovered this called that RNA replicase. And it is the enzyme that the early experimenters in RNA evolution decided to use to allow RNA to replicate. We now have a bunch of different ones that we use and a bunch of different chemistries that we use. Again, we're trying to get chemistries that are more and more similar to chemistries that would be found on the early Earth. But in these early experiments, we were using these complex proteins to allow RNA to evolve. And that complex protein came from this virus. So this first experiment, go back to my paper here. This first experiment, just what he did is he set up some test tubes with a really simplified chemistry of something that you would find somewhat similar to inside of a bacterial cell. So he set up the salinity to be similar, and a lot of the he loaded it with RNA nucleotides, so single monomers of RNA, and he loaded it with chains of RNA that had that specific viral sequence that allowed them to be recognized by the RNA replicase, the protein that allows those RNA chains to reproduce. And then he inserted the RNA replicase. And what he did is he just let them replicate rapidly. And then he would take a small sample of the liquid that was produced, of the solution that was produced, and he would squirt that into a new vial and let things replicate in there. And he just kept doing this over and over and over again. He did that for 40 times, 40 inoculations is what they call it, when you inoculate a nice clean batch of material with your original evolving strain. And after 40 generations, if you think about what's happening here, the natural selection going on in this experiment, the nature here, which is, of course, the inside of this test tube, that environment is selecting for chains of RNA that are better and better at attracting that RNA replicase protein and replicating rapidly and then doing it again. So he was selecting for super rapid replication. And what happened is that after just 40 transfers, his molecules were replicating 15 times faster than the original molecules from the virus. So he had rapid evolution towards really fast replication. And when he looked at the structure of these RNAs to see what it was that they did, how did they evolve this super rapid replication, it was almost disappointing how simple it was. What they did is they just, any mutation that made the RNA shorter without ruining the stretch of RNA that attracts the replicase, any mutation that made them shorter made them replicate faster. And so not only were they 15 times faster, they were also 15 times shorter than the original nucleotides. And at first, if we're thinking, we want this process to be able to give rise to new complexity, all he did was make something shorter. How is that evidence of evolution here, right? But he pointed out in his first paper, he writes, it should not escape the attention of the reader that other selective stresses, aside from rapid replication, other selective stresses can be imposed on the system to generate RNA entities which exaggerate other molecular features. So in other words, if we put obstacles in the way of replication, these RNA chains should evolve new functions. They should evolve new complexities. And by the way, experiments showed that they did. But this brings me to kind of a weird point that I want to talk about here. Real quick, a quick aside. There are two ways of existing in the universe. One way is to be really hard, like a rock, in your environment. And if you are hard, like a rock, you're going to persist in your environment, right? So that's one way that things can exist in our universe. They can be hard. They can be really stable within their environment. Another way to be common in the universe is to just get reproduced or produced really easily. So snowflakes on Earth are a common example of something that is fragile. It doesn't exist very long, but they get produced so often that they are actually really common on planet Earth. Snowflakes are very common on planet Earth. So there are these two ways to exist. Living things are kind of a combination of both. We replicate rapidly, and we're also fairly resistant to degradation, right? And different organisms have evolved. Some have evolved to replicate super quick, but they're also very fragile. And his little RNA chains that he created through evolution in this evolution experiment, they are examples of these organisms that reproduce extremely rapidly, but are very, very fragile. Pretty much any obstacle that they come across, they will die. These really simplified molecules that he ended up evolving in there, but they replicate so rapidly that that's okay, right? We are examples of organisms that do both. We're very stable. You know, in my first live podcast, I mentioned that we're sort of just an extension of our balls or our ovaries. You know, we're all about replication. It's not totally true because we're also about survival. We are very stable organisms, and we wait for the right time to replicate. And so basically what he was saying, that after doing this first RNA replication experiment, he was saying that in future experiments, we should give the RNA some obstacles. We should give it a reason to wait before it replicates, do other things besides replication. And he believed that we could get it to evolve complexity. And sure enough, he was right. In section 3 of this paper, he goes on to outline some other experiments that he did, where he would add weird obstacles to these RNA chains as they were trying to replicate. So one of the things that he did is he would starve them of nutrients. You know, the types of nutrients that these chains need, that they need RNA nucleotides in order to replicate. That's what they make their bodies out of. And they need other molecules, and he just deprived them of some of the molecules that they needed for replication. And if he deprived them all at once of those molecules, they would die. So the lineage would go extinct. But if he slowly deprived them of those molecules, they would evolve and adapt. Their sequences would change through descent with modification, acted upon by selection. Their sequences would change, they would adapt, and they would be many times more efficient at survival and reproduction without the molecules that the original chains of RNA needed to replicate. He also added some toxins, things that got in the way of, specifically of base pairing. And again, it was the same thing. If you added too much of the toxin right away, the thing would go extinct. If he added it slowly, the things would adapt. They would actually evolve to the obstacles that he put in their way. Just, it's so cool. These experiments were so awesome. He's doing all of this just with RNA replicating. Again, there's no genetic code. There's just, he had to set up a very specific environment for them, but there was no genetic code needed. No genetic code needed for this evolution to occur, and no cells needed. So the final thing that started happening is we realized, look, these things are evolving new traits, new abilities in ways that we can't even, we have to then study them to figure out how it was that they solved the problems that we threw at them. Evolution is coming up with very sophisticated solutions to the problems that we throw at it. I wonder how crazy we can get. The other big experiment that they did is they threw in a predator, an RNA predator. They threw in an enzyme called RNase. And this particular RNase, it digested RNA by grabbing the U nucleotides and the let's see, what was it? The C nucleotides and the U nucleotides. If any of those nucleotides were loose on an RNA chain, this enzyme would grab the RNA chain and start to digest it. So it was an RNase. It's an enzyme that hunts down chains of RNA and starts degrading them by grabbing the C or U nucleotides that are loose and starts digesting them. And he found this structure right here that we see on the page. I'm not going to go into the detail about how this works, but it evolved a way to hide all of its loose C and loose U nucleotides. Its structure evolved in a way that could hide from this predator, and it could still replicate. This is so cool. You have a predator that's just made out of protein. It's not actually a full-on living organism like we would consider today. And you have a chain of RNA that's also, we wouldn't really consider it a full-on living organism, but it's acting like an organism. It's evolving ways to avoid a predator. This is so cool. Just all in the test tube, just right before our very eyes, all of this happened. And we know the actual sequence. We actually know the trick that RNA chain evolved to avoid its predator. This is just so cool. Then to top all of this, researchers said, you know what? Evolution is so good at finding novel solutions to problems. What if we trained through this evolutionary process a chain of RNA? What if we evolved a chain of RNA that could do complex medical things for us? What if we evolved a molecule that could bind specifically to a protein in the body that's problematic and use it as a medicine to actually fix a real problem? And sure enough, we were able to do this. There is a disease that causes blindness in old people. So hold on, let me just read my notes here because there's a lot of vocabulary here that I'm not familiar with. So a process was used to invent a medication, Mucogen, that's the name of the medication, which stops the progression of a specific type of macular degeneration. So macular degeneration is a type of blindness that elderly people get. It's, adult onset blindness. And they actually evolved a molecule that it can't restore sight, but it can stop the progression of blindness in elderly people. Again, it has to be a specific type of this disease, but it can actually stop the progression of this blindness. And it's a molecule that we evolved through evolution, RNA evolution experiments. Since that, this was one of the earliest molecules that was evolved, the earliest medications that was evolved in this way. Since then, we've done very similar things to evolve all sorts of different types of molecules. Imatinib is a molecule that was also evolved, not through a process exactly the same. We used a selection process. We built this machine that could mutate molecules that aren't chains of RNA, can mutate pretty much any type of a molecule. And it would just, through an evolutionary trial and error process, it would just change a molecule slightly, then test that against its medical target, then mutate it again slightly, test it again, it's tested again against its target, and so on and so forth, until we found imatinib, this molecule that's used to treat chronic myelogenous leukemia, a very specific type of leukemia. And just recently, we started using this to treat leukemia years ago. And just this year, a paper was published saying that imatinib, not only does it treat this type of cancer, it can actually cure it. People who have been using the medication for 5 to 10 years, a lot of times when we take them off the medication, we find that the disease has been completely cured. We have now used evolution to cure cancer. All right. Darwin's theory of evolution has literally allowed us to heal the sick, give sight to the blind, or at least stop the progression of blindness, and cure cancer, right? This is insane. So anyway, very impressive, these experiments. I just want to end things here by opening up to questions once more. If people have any. And again, super chats are welcome, folks. This is the time of the podcast where I play the organ music and pass around the collection plate, on this Sunday morning science broadcast. Brian Stevens, are you asking a question there? Assume you, Ian. Yeah, rewrite that, please. Someone says, is it true that we can eventually slow down or reverse the aging process? Maybe. The problem with aging is we don't have a good theory of aging that is fully worked out. A lot of people think that aging has to do with avoiding cancer. We start killing cells when they start accumulating mutations. Cells will commit suicide as they accumulate mutations in order to avoid producing cancer. And this process is one of the things that we think causes aging. And in order to kind of reverse engineer that is going to be extremely complex. So a lot of the if we want to actually edit our genes and edit mutations, for example, that crop up in our bodies as we're growing and maturing, we have to find a way to edit all of the cells in our body at the same time. We need a delivery system to deliver things like, if we end up using CRISPR to do it or whatever. And these delivery systems are really hard. That's one of the big holdups that we're having. So even if we do really understand aging to the point to where we could correct it, we also need to find a way to actually, deliver gene editing technologies to all of the cells in our bodies. So it's probably possible, hypothetically, but we're a long way away from it. And there's going to be a lot of problems with it, I'm sure, that we'll have to work out. Okay, Brian Stevens says, assuming you mean topoisomerase 2 inhibitors as a leukemia killer. Imatinib is actually a really small molecule. It's a tiny little molecule that goes in. It gums up a protein that is a result of the mutation that gives rise to this specific type of myelogenous leukemia. So there's a chromosome fusion in this type of leukemia that creates a really weird protein, and this molecule gums up that protein. Whether or not that's the protein that you said there or not, I don't know. I'd have to look that up. But yeah. Someone says, when we'll be able to cure low IQ? Asking for a friend. Yeah, I don't know. It looks like that is kind of it for questions. Hopefully everybody was able to follow this conversation. I know it was sort of difficult if you haven't watched the RNA videos or thought much about RNA replication or the RNA world hypothesis, but this is really cool stuff. Really, really neat stuff. The research that's been going on in this is absolutely insane. The things that we've been able to do since understanding how life works, how genes work, is really, really amazing. Someone asks, can you explain why RNA is the most plausible hypothesis? RNA is not necessarily the most plausible hypothesis, but it's the route that it's the molecule that it looks like was really important for several reasons. One is that the ribosome, which is this really complex molecule that takes chains of RNA and reads them out into protein. The ribosome is made of RNA. So unlike other enzymes in our body, which are made of protein, it's made of RNA, and it's one of the most fundamental, molecules in our body. It does have some protein in it, but the proteins in the ribosome are just used to stabilize the molecule. All of the functional bits of the ribosome are made of RNA. So that was one of the big clues. The other big clue is that RNA can act both as a reproducing molecule. It can act as a template for its own replication, and it can also fold up and act as an enzyme. And it's that dual property of a single molecule that makes it really promising for the original evolving entity. Now, RNA itself is fairly complex. Its nucleotides are fairly fragile, and it has to be in chain-like form in order to start to replicate, right? And it also needs an enzyme that can help it replicate, or catalyst that can help it replicate. So we don't think that it was the original thing that evolved. There's a separate type of evolution called chemical evolution, which works a little bit differently. There's not descent with modification so much as there is repetitive production and an evolving chemical system. And I would need another cup of coffee to go into that right now. That's kind of a topic for another day, but there's a really good paper. I'll link to it in the video description on the metabolism first hypothesis. So it's believed that a metabolism actually evolved, a metabolic system evolved first. That gave rise to RNA, which could actually replicate through this template directed replication. That type of replication eventually gave rise to the ribosome, and eventually we get living cells and everything from that today. So, but yeah, another topic for another day. Someone asks, so if it's not the most plausible, then is it because of the dual purpose that makes the hypothesis, that makes it the hypothesis that scientists mainly subscribe to? I wouldn't say it's the hypothesis that people mainly subscribe to. It's the one that's getting the most research done. And it's for multiple reasons. First of all, is that the dual properties of RNA, but also the fact that RNA is used in every single organism today and in every virus. It's super prevalent in life today. The reason that I say it's not the only plausible solution, or necessarily the most plausible solution, is because it is a complex molecule. Even its monomers are complex, and they're not very stable in environments that we would imagine existed on the early Earth. But there are, so Nicholas Hudd, who works at the Center for Chemical Evolution, he is studying alternatives to RNA that are similar to RNA but are more stable and more readily produced in an early Earth environment that could have transitioned into RNA. So there's that type of research that's happening too. That's why I say it's not necessarily the most probable. But all of the experiments that we do on how early evolution would have happened, we use RNA because, again, it's in cells today. It's what the ribosome is made of. And we already have all this technology for sequencing chains of RNA. We already have all the equipment to study RNA. So there's lots of reasons RNA is used in life origins research. J.R. Pone did a super chat. Thank you. Just want to say I love your channel and live topics. So cool. Thanks. I appreciate it. We got one more question. Is it possible that we could evolve RNA that could evolve without a catalyst? I think if you slowly remove the catalyst from the environment, it might be able to happen. You do need a catalyst, but there's some newer research. And if you look at this the metabolism first hypothesis, they're looking at really, really simple catalysts. So in experiments that we learned about today, we're using a complex catalyst that's a protein. It's an enzyme. But much simpler catalysts can also do the job given the right environmental circumstances. And if we have an evolving metabolic system, we could get something like this to happen. There are also experiments on enzymes made of RNA itself. So and actually this paper, the 40 years of evolutionary experiments, that paper actually goes on to talk specifically about the evolution of RNA enzymes. So a chain of RNA that can replicate other chains of RNA. It's that backbone binding process that's hard to do. And people are trying to evolve these RNA chains that can do that. But it's kind of a chicken and egg scenario. You could evolve an RNA chain that can do that, but you needed to use evolution to produce that. And you can't get evolution to happen without the replication. So this metabolism first study is really, I think, where the money's at for understanding the origin of life, understanding the origin of the RNA world, right? So there's that. Okay. I'm going to close things out with that. Thank you for watching. This has been a lot of fun. Hopefully you enjoyed going through this paper. If you'd like me to if you have ideas for future topics, let me know. I think that next week I'm going to talk about the evolution of mimicry. I just produced a video for the Stated Casually channel on snake mimicry, which you should watch. That was just published yesterday. And yeah, so long for now. Stay curious. It's been a lot of fun.